In all living systems, the genetic information is encoded by a simple code of 4 nuclear bases, A, T, G, and C. The human genome contains 3 billion base pairs of this chemical code. But did you know that nearly 2% of all bases in the DNA are structurally modified? My research examines both naturally occurring DNA modifications that are introduced as part of epigenetics control, and also toxic and mutagenic DNA adducts that are created as part of DNA damage. We strive to identify the structures of novel DNA modifications, to examine their effects on biological processes, and also to develop small molecule inhibitors that can influence the processes of epigenetic programming in cells. Unintentional DNA modifications that occur as part of DNA damage by environmental chemicals, dietary components, and endogenous reactive molecules can contribute to cancer and aging. In fact, we know that DNA damage accumulates in the brain and in the heart with age. So my laboratory discovers the structures of DNA modifications that are formed in our genome and investigates their biological consequences, such as their effects on DNA replication, transcription, and repair. As part of this process, we employ the tools of organic chemistry to synthesize unnatural DNA nucleosides and incorporate them into synthetic DNA strands using solid phase synthesis. We then employ biochemical experiments with the synthetic DNA strands in order to elucidate their biochemical outcomes. Our bodies are composed of over 120 cell types, which differ dramatically in terms of their morphology and function, but carry exactly the same set of genetic instructions. This difference between cell types is imposed by epigenetic control, which influences how the DNA is packaged within the nucleus, making some genes available for transcription and others hidden within the chromatin. While epigenetic marks are very stable in healthy somatic cells, they are completely scrambled in tumors. Specifically in tumors, we see increased levels of DNA methylation and decreased levels of DNA hydroxymethylation. My laboratory investigates the way that epigenetic control occurs in cells, specifically on the level of DNA, by introducing and removing specific epigenetic marks. And we are also working on development of epigenetic inhibitors that can influence the way the epigenetic marks are introduced. These epigenetic inhibitors are useful for understanding the functions of various epigenetic modifications and also as potential therapeutic agents for treatment of cancer and affecting other diseases which are influenced by aging. My laboratory contains subgroups dedicated to organic synthesis. This includes solid phase synthesis, cell culture, and molecular biology, and also advanced analytical chemistry. We have 2 mass spectrometers that are located in our laboratory, and all graduate students and postdocs have a chance to develop skills in operating and using these experiments, as well as develop analytical methods for sensitive and specific detection of DNA modifications. Many graduates choose to join biotech companies such as Genentech, BMS, Pfizer, and many other leading pharmaceutical companies in the US and abroad. Our studies of naturally occurring epigenetic modifications of DNA are significant because they reveal the natural mechanisms of cell differentiation, carcinogenesis, and they also enable us to develop novel therapeutic agents that target DNA processes. Our research in the area of DNA addicts is important because it makes it possible to understand the specific biochemical mechanisms by which chemical damage can be transferred into genetic damage and lead to the initiation of tumors. Accurate measurements of the amounts of DNA addicts present in tissues and biological liquids help us identify individuals at risk and also to use these DNA addicts as biomarkers to establish the efficacy of anti-tumor drugs. Research in my laboratory is highly interdisciplinary and it crosses many boundaries between organic chemistry, biochemistry, advanced analytical chemistry such as biological mass spectrometry, as well as molecular and cell biology and epidemiology. My work has been published in a range of journals, including Angewandte Chemie, Journal of the American Chemical Society, Nucleic Acids Research, Chemical Research and Toxicology, and others. I hope you will consider joining my group at University of Minnesota. You will learn many new skills, including nucleoside, nucleotide, and oligonucleotide synthesis, structural characterization of novel nucleosides, advanced mass spectrometry techniques, HPLC, as far as human cell culture, gel electrophoresis, and other biochemical techniques. You will also develop skills in the fields of experimental design, manuscript and grant proposal writing, oral presentation, and will have a chance to mentor an undergraduate or two during your career here as a graduate student. You will work in a collaborative environment where students from 3 different graduate programs in medicinal chemistry, chemistry and biochemistry work together on common goals in the areas of chemical carcinogenesis, biochemistry, and drug design. Thank you for considering my group.